Your body is destroying itself right now, and the moment you leave Earth, it starts killing you faster. Tonight, we're going to talk about something the space industry doesn't want to say out loud. The problem with reaching the stars isn't the rockets, it isn't the fuel, it isn't even the distance. The problem is you. The problem is this fragile biological machine you live inside. And by the end of tonight, you're going to understand exactly why the human body is the single greatest obstacle standing between our species and the cosmos. Before we get started, if you love exploring the depths of space as much as we do, take a second to like the video or subscribe. It's a simple action, but it helps this channel reach more curious minds like yours. Now let's begin. There is a moment in every serious conversation about interstellar travel where the numbers stop adding up. Not the engineering numbers, not the fuel calculations, not the propulsion equations, the biological numbers, the ones that describe what space actually does to the machine we call the human body. And when you look at those numbers carefully, when you stop celebrating the idea of reaching the stars and start actually measuring what it would cost the people doing it, something deeply uncomfortable becomes clear. We are not built for this, not even close. We are built for a specific gravity, a specific radiation shielding, a specific atmospheric pressure, a specific magnetic field, a specific temperature range, a specific oxygen concentration. We are built for one planet. We evolved here over millions of years in response to one set of conditions. Earth didn't just create the environment we live in, Earth created us. And when you remove us from Earth, even partially, even temporarily, the body begins to fall apart in ways that are not minor inconveniences to be engineered around. They are fundamental, deep, systemic failures that cascade through every organ system, every cell, every molecule. And we know this not from theory, we know this from data, from decades of space station research, from 17,000 hours of cumulative human spaceflight, from blood samples and bone scans and eye exams and brain MRIs taken before, during and after missions, from astronauts who are among the healthiest, most carefully selected and meticulously trained human beings who have ever lived, who spent months in orbit and came back measurably broken in ways that took years to partially repair. And those astronauts were in low Earth orbit, still within the partial protection of Earth's magnetic field, still reachable by emergency return vehicles, still receiving resupply missions every few months, still able to call home with a delay of less than a second. Now imagine the same biology, the same fragile Earth-adapted body, crossing 424 light years to reach Proxima Centauri. 25 trillion miles of unshielded deep space, away from every protective field our planet provides, at the absolute fastest speed any craft we can currently imagine might achieve, not for 6 months, not for a year, for decades. The math does not work, not because the distances are too great, though they are, not because the fuel requirements are prohibitive, though they are, but because the human body would come apart long before it arrived. This is not pessimism. This is not a failure of imagination. This is what the science says very clearly when you read it without the filter of wishful thinking that has always colored our relationship with the stars. Tonight, we're going to walk through exactly what happens to a human body in deep space, system by system, cell by cell. We're going to look at the radiation environment that exists beyond Earth's protection, what it does to DNA, what it does to the brain, what it does to the cardiovascular system, what it does to the immune system. We're going to look at what happens to bone and muscle in extended microgravity, and why 6 months of data from the International Space Station suggests that years in zero gravity might be a threshold the human body simply cannot cross and recover from. We're going to look at what happens to vision, to fluid distribution, to the pressure inside the skull when you remove the gravitational gradient we evolved to live within. We're going to look at what happens to the human mind in total isolation over years and decades, with no possibility of rescue, no natural environment, no sensory variety, no physical connection to the world that built us. And we're going to look at reproduction and what space does to the biological machinery that creates the next generation, because any serious interstellar mission involving a generational ship would have to solve that problem too, and it has not been solved. And then, having laid out all of that, we're going to talk about what might actually happen, not in 50 years, not in 100, but in the real long arc of time. If our species lasts long enough and becomes capable enough, what would a genuine interstellar mission look like? What would have to change about us? Would those changed beings still be us? And what does it mean to reach the stars if the things that arrive aren't the people who left? These are not comfortable questions, but they are the right ones. Let's start at the beginning. Let's start with the most immediate, the most constant, and arguably the most lethal threat that space poses to any biological organism that dares to leave Earth's protection. Let's start with radiation. Imagine walking outside every morning, and instead of feeling the warmth of the sun on your face, you walked into a shotgun blast, not metaphorically. A genuine shotgun blast of subatomic particles moving at percentages of light speed. Particles with so much kinetic energy that when they hit the atoms in your cells, those atoms shatter. The electrons get ripped away, the bonds between molecules break. The DNA inside your cells, those delicately coiled ladders of genetic information that took 3.8 billion years of evolution to write, gets hit directly. Not just the outer coating of the cell, not just the membrane or the cytoplasm, the actual genome, the actual blueprint. This is what happens in deep space every single second. On Earth, we are extraordinarily well protected from this. We live at the bottom of an ocean of air, roughly equivalent in shielding to 9 feet of concrete in terms of stopping radiation. We also live within a magnetic bubble, the magnetosphere, generated by Earth's molten iron core spinning deep beneath our feet. That magnetic field deflects charged particles from the solar wind, bending their paths around the planet like water around a rock. Together, these two shields reduce our radiation exposure to a background level of about 0.3 rem per year. That's enough to be essentially harmless over a normal human lifetime. On the International Space Station, that changes. The station orbits at about 250 miles altitude, still within Earth's magnetic field but above most of the atmosphere. Astronauts there receive about 15 to 20 times the radiation of someone on Earth's surface. For a 6-month mission, that adds up to roughly 80 millisieverts. This is manageable, though not trivial. It increases cancer risk over a lifetime in ways that NASA and other space agencies carefully monitor and regulate. There are career dose limits for ISS astronauts. There are guidelines about how many missions someone can fly. The risk is understood, it's accepted, and it's mitigated as best as possible within the constraints of a vehicle that still sits within the Earth's magnetic bubble. Now travel beyond that bubble, now go to deep space, which means anything beyond about 60,000 kilometers from Earth, where the magnetosphere fades and the interplanetary medium begins. There, 2 entirely different radiation environments wait for you. The first is solar energetic particles. These are bursts of protons and electrons ejected from the sun during solar flares and coronal mass ejections. They arrive with relatively little warning, typically hours at most. During a major solar particle event, an unshielded astronaut in deep space could receive a lethal radiation dose in minutes. Not the kind of dose that increases cancer risk years later. A dose that causes acute radiation syndrome within hours. Nausea, vomiting, hemorrhage, immune system collapse, death. The astronauts of Apollo 16 and Apollo 17 were extraordinarily lucky that no major solar events occurred while they were on or near the Moon. The large solar particle event that occurred in August 1972 between Apollo 16 and Apollo 17 would have been fatal to a crew in deep space without adequate shielding. They missed it by months on either side. You can shield against solar particles to a reasonable degree with sufficient mass around the crew compartment. Water, polyethylene, and other hydrogen-rich materials are reasonably effective at absorbing these lower-energy protons. For a spacecraft with enough mass, solar particle events are survivable. They are a solvable engineering problem, though every kilogram of shielding adds to the launch mass and thus the fuel requirements of a mission that is already, as we will discuss, nearly impossible to fuel. But the second radiation environment is not solvable in the same way. The second radiation environment is what keeps physicists and biologists awake at night when they think seriously about interstellar travel. Galactic cosmic rays. These are not from the sun, these are from dying stars, supernovae, active galactic nuclei, neutron star mergers. They are the most energetic subatomic particles that exist in the natural universe. They are primarily protons and the bare nuclei of heavier atoms, iron, silicon, carbon, traveling at anywhere from 90% to 99.99999% the speed of light. They have been accelerated over millions of years by the shock waves of stellar explosions and the magnetic fields of dying stars. Some of them carry more kinetic energy in a single particle than a well-thrown baseball. They come from every direction simultaneously. There is no point in space where you are shielded from them by the interstellar medium, because the interstellar medium is nearly empty. There is no time of day or solar cycle phase where they diminish significantly. They are constant, omnidirectional, and nearly impossible to stop. And here is the crucial physical fact that makes galactic cosmic rays so uniquely threatening. When a proton moving at 99% the speed of light hits a thick lead wall, the proton does slow down and stop, but the collision produces a cascade of secondary particles, neutrons and gamma rays and pions that actually penetrate further into the material than the original proton would have. This is called shielding-induced secondary radiation. It means that for galactic cosmic rays, adding more physical shielding up to a certain point actually makes the radiation problem worse before it makes it better. You would need to surround a spacecraft with roughly 2 meters of water or the equivalent before you would begin to see net attenuation of galactic cosmic ray damage. 2 meters of water surrounding an entire crew compartment on an interstellar spacecraft. The mass required for that kind of shielding is staggering. The kind of mass that would require energy sources and propulsion concepts we have not yet invented. NASA estimates that for each year an astronaut spends in deep space beyond the magnetosphere, approximately 1/3 of their DNA will be hit directly by galactic cosmic ray heavy ions. 1/3 of the genome struck directly in a year. Not by the relatively low energy radiation we use in cancer therapy or that you might receive during a CT scan, by particles that smash through biological tissue like bullets through tissue paper, leaving long trails of ionization in their wake, tracks of damage that can span entire cell nuclei. The effects are not limited to cancer, though cancer risk alone is severe. At sufficient cumulative doses, galactic cosmic rays cause what researchers call central nervous system effects. This means measurable, lasting changes to how the brain functions. Studies in mice exposed to simulated galactic cosmic ray radiation have shown significant impairments in memory, spatial navigation, and decision-making. The cognitive performance of irradiated animals declines in ways that do not fully recover over the animal's remaining lifetime. Fear responses become dysregulated. The animals become anxious and prone to freezing behaviors. The damage appears to involve direct injury to neurons in the hippocampus and prefrontal cortex, the regions most critical for the kind of complex reasoning, problem-solving, and emotional regulation that a multi-year mission would demand. These are animal studies We cannot directly extrapolate to humans, but the neuroscience community does not find the prospect comforting. Research published as recently as 2025 from the Lawrence Berkeley National Laboratory has examined the mechanisms by which galactic cosmic ray components induce cognitive damage in animal models, and the picture is not a comforting one. Radiation of this type causes chronic neuroinflammation. It damages the myelin sheaths around nerve fibers. It disrupts synaptic connectivity. These are not effects that cells can simply repair and move on from the way they repair the damage from a brief X-ray. They are cumulative, they worsen over years, and they have no known effective countermeasure. Then there is cardiovascular damage. Studies of atomic bomb survivors and patients who received chest radiation for cancer treatment have established a clear relationship between ionizing radiation exposure and long-term cardiovascular disease. Radiation damages the endothelial cells lining blood vessels. It accelerates the formation of atherosclerotic plaques. It causes inflammation in cardiac tissue. It increases the risk of arrhythmias. For astronauts on multi-year deep space missions, the cardiovascular consequences of radiation exposure may be as serious or more serious than the cancer risk. This is an area of active and urgent research, particularly as planning for crewed missions to Mars has become more serious, and researchers are finding that our understanding of radiation-induced cardiovascular disease remains significantly incomplete. There is also the immune system. Radiation suppresses the production of white blood cells by damaging stem cells in the bone marrow. During extended deep space missions, radiation could reduce immune competence to levels that would make infections that are normally trivial become life-threatening. This is compounded by the fact, documented in ISS research, that spaceflight already appears to reactivate latent viral infections. Astronauts on orbit have shown reactivation of Epstein-Barr virus, varicella-zoster virus, and cytomegalovirus, all of which remain dormant in most healthy people on Earth but begin replicating in the immunosuppressed environment of space. In deep space, with no medical evacuation option and limited medical resources, a reactivated infection or a novel infection in an immunocompromised crew could be catastrophic. Perhaps most disturbing of all are the effects on the genome itself, not just through direct DNA strand breaks, but through epigenetic changes. NASA's famous twins study, which followed astronaut Scott Kelly during his year on the ISS while his identical twin brother Mark remained on Earth, found that spaceflight caused widespread changes in gene expression. Genes involved in immune function, bone metabolism, and DNA repair were significantly dysregulated. More disturbingly, when Scott returned to Earth, while most of these changes reversed, roughly 7% of his gene expression remained permanently altered. 7%, after 1 year, mostly within Earth's protective field. The study's authors noted carefully that the long-term health consequences of these persistent changes remain unknown. For an interstellar mission, these changes would not be measured in percentages of one genome over 1 year. They would accumulate over decades. The crew arriving at their destination, if they arrived, might be genetically and epigenetically so different from the humans who departed that the question of whether they are still the same people becomes not just philosophical, but biological. The radiation problem alone, considered seriously, represents a barrier that no current or near-future technology can adequately address. It is not a problem that better spacecraft design fully solves. It is not a problem that better drugs fully solve. It is a physical reality of the deep space environment, one that arises from the fundamental violence of the universe. Stars explode. They produce energies that would be lethal to life. The interstellar medium is bathed in the remnants of those explosions. Any biological organism attempting to traverse that medium will absorb those remnants. The cells will be damaged, the genome will be hit, the brain will be affected, the cardiovascular system will suffer. And with no protective planet beneath you and no magnetic field around you, there is nowhere to hide. But radiation, as devastating as it is only the beginning. Because even in a perfectly shielded spacecraft, even in a vessel so massive and well-designed that galactic cosmic rays were somehow rendered harmless, the human body would still be destroying itself slowly and systematically through a second mechanism that is just as fundamental and just as difficult to counter. Gravity, or more precisely, its absence. You have never felt gravity? Not really. Not as a thing separate from yourself, the way you feel heat or pressure or light. Gravity is so constant, so omnipresent in your experience, that it is indistinguishable from the baseline of existence. You wake up in it, you eat in it, you breathe in it, your heart beats in it, your bones are shaped by it, your muscles exist in their current form because of it, your circulatory system pumps blood in a direction determined by it. Your inner ear interprets the world in terms of it. Every organ in your body, every tissue, every cell has been shaped over hundreds of millions of years of evolution to function correctly in the presence of a specific gravitational force. 9.8 meters per second squared. Earth's surface gravity. Not Mars's gravity, not the Moon's gravity, not the 0.38 g of Mars, not the 0.165 g of the Moon. 1 g, the number that Earth has provided consistently for the entire history of life on this planet. When you remove that gravity, the body does not simply float neutrally and continue functioning. It begins to break down, not dramatically, not immediately, but persistently and cumulatively in ways that become genuinely frightening when you extend them over the timescales required for interstellar travel. Let's start with bone. Bone is not a static material. It is a living tissue constantly being remodeled by two competing cell types. Osteoblasts build new bone. Osteoclasts break old bone down. On Earth, the mechanical loading of gravity on the skeleton keeps this process in balance. Stress on a bone from bearing weight against gravity is the primary signal that tells osteoblasts to keep building. Without that signal, the balance tips dramatically toward resorption. The osteoblasts slow down, the osteoclasts continue at or increase their rate. The result is a net loss of bone mass. We have been measuring this process in astronauts for decades. The numbers are consistent and alarming. In microgravity, despite extensive countermeasure protocols involving hours of daily exercise on specialized resistance devices, astronauts lose bone mineral density at a rate of approximately 1 to 1.5% per month in load-bearing bones, the lumbar spine, the pelvis, the femoral neck of the hip. These are the structural bones most critical to standing, walking, and bearing the weight of the body against gravity. The bones that need to work when you arrive at a destination and try to stand up. For a 6-month mission, that means losing roughly 6 to 9% of load-bearing bone density. This is equivalent to about a decade of normal age-related bone loss in a healthy adult, compressed into half a year. Astronauts who have completed 6-month ISS missions are effectively returning to Earth with the skeletal system of someone significantly older than they left. And the recovery is incomplete. A study published in 2022 in Scientific Reports examined bone microarchitecture and strength in 17 astronauts before flight, at return, and at 6 and 12 months post-flight. 12 months after landing, multiple measures of bone strength, density, and microarchitecture in the tibia remained significantly reduced compared to pre-flight values. Astronauts who flew longer missions showed less recovery than those who flew shorter ones. The damage was not fully reversible within a year. A follow-up study published in 2025 looked at 2 astronauts 4 years after long-duration spaceflight. One had achieved full recovery. The other still showed trabecular deficits at that 4-year mark, partially compensated for by cortical thickening, but not fully restored to pre-flight architecture. Extend this to an interstellar mission, even a transit to Proxima Centauri at a hypothetical 10% of light speed, which would require propulsion technology we do not have, and fuel quantities we cannot currently produce would take 42 years. 42 years of microgravity transit. Even with the best exercise countermeasures we could design, the bone loss over that period would likely be catastrophic and irreversible. The bones would become so fragile that the simple act of landing at the destination and experiencing full gravity for the first time in decades might fracture them. Not because of a fall or impact, but because the structural material was no longer sufficient to bear the body's own weight in a gravitational field. And that assumes we maintain 1g at the destination. We don't know the gravity of any exoplanet we might want to visit. We don't know if Proxima Centauri b, if it's habitable, has enough mass to provide adequate gravity. We know its minimum mass, which is roughly Earth's. But the actual mass, and thus the actual surface gravity, could be quite different. If the surface gravity is less than Earth's, bone loss would accelerate. If it's greater, an already weakened skeleton would bear a disproportionate load. Bone loss is only the beginning. Muscle atrophy in microgravity is equally severe. The postural muscles, those that maintain your spine upright, hold your head in position, keep your core stable, simply stop working in weightlessness. There is no posture in microgravity. There is no upright to maintain. Within weeks, without aggressive exercise, the postural muscles begin wasting at visible rates. Even with the 2 to 2.5 hours of daily exercise that ISS crews perform, a 6-month astronaut returns to Earth having lost 20 to 30% of lower limb muscle mass and significant core strength. They are typically unable to walk unassisted for days after landing. They require rehabilitation programs lasting months to regain full physical function. Again, extend this over 42 years. Even with the most effective exercise interventions we can design, keeping the musculoskeletal system functional for decades in weightlessness is not a solved problem. It may not be a solvable problem. The maintenance of muscle mass requires not just mechanical stimulation, but the hormonal and metabolic context that comes with living in a gravitational environment. Growth hormone release patterns, cortisol rhythms, testosterone, and estrogen levels. All of these are influenced by the body's gravitational state, and all of them shift in weightlessness in ways that are not fully compensated by exercise. The cardiovascular system undergoes its own profound changes. In microgravity, the hydrostatic pressure gradient that normally drives blood preferentially downward toward the legs disappears. Roughly 2 liters of fluid, blood, and interstitial fluid shifts from the lower extremities toward the head and thorax. In the first days of spaceflight, astronauts experience facial congestion, stuffy noses, elevated intracranial pressure, and a sensation not entirely unlike being permanently slightly upside down. The body interprets this fluid shift as a sign of fluid overload and begins excreting it through the kidneys, reducing total blood volume by about 15%. The heart, no longer working against gravity to push blood upward, begins to deconditioning. It gets slightly smaller. Its maximum output capacity decreases. The blood vessels in the lower legs, which on Earth must maintain tone to resist the pooling of blood under gravity, lose their tone in weightlessness because they don't need it. The autonomic nervous system recalibrates to a low-gravity state. When these astronauts return to Earth, the cardiovascular deconditioning is immediately apparent. The heart cannot adequately pump against gravity. Blood pools in the lower extremities. Blood pressure drops when the person stands. This is called orthostatic intolerance, and it is severe enough after long missions that some astronauts experience near-fainting episodes upon landing. The cardiovascular system has to completely recalibrate to operate in a gravitational field again. This process takes weeks to months and is often incomplete. For 42 years of microgravity transit, the cardiovascular recalibration required upon arrival at a destination with a gravitational field might exceed what the system is capable of performing. The heart might simply be unable to pump adequately against gravity after decades of deconditioning. The blood vessels might lack the structural tone to maintain appropriate distribution. The person might arrive in a state of cardiovascular failure, not because of any disease process, but because the hardware was optimized for a gravity-free environment over such a long period that it lost the ability to function in any other way. Artificial gravity is, of course, the theoretical solution. If you spin a spacecraft, centrifugal force mimics gravity. The required spin rates for comfortable, Earth-equivalent gravity require a rotation radius of at least 100 meters before the Coriolis effect, that disorienting sideways tug that occurs when you move relative to the spin axis, becomes tolerable. This means an interstellar spacecraft would need rotating sections roughly 200 meters in diameter. This is not impossible in principle, but it is enormously costly in mass and engineering complexity, and it does not solve the radiation problem, and it does not solve the psychological problem, and it does not solve the immune problem, and it does not solve the reproductive problem. It is a partial solution to one aspect of one of many problems, and it comes at a cost that compounds all the other costs. But let's set aside for a moment both radiation and microgravity. Let's imagine a hypothetical spacecraft that has somehow solved both those problems. The crew is in perfect Earth-equivalent gravity. They are behind shielding that somehow stops galactic cosmic rays. Now, what happens to them over 42 years? There is a condition that appears in about 60% of astronauts on long-duration space flights. It doesn't get discussed as much as bone loss or radiation, perhaps because it doesn't fit neatly into the heroic narrative of space exploration. It's called Spaceflight-Associated Neuroocular Syndrome, or SANS, and it may be the most quietly alarming thing we have discovered about extended human presence in space. When you spend months in microgravity, the fluid that normally distributes itself under gravity, with more in the lower body and less in the skull, redistributes toward the head. This is the same fluid shift that causes facial puffiness and nasal congestion in the early days of a mission. But beyond the discomfort of the early adaptation, the sustained elevation of cerebrospinal fluid pressure in the skull has more serious consequences. The optic nerve, where it exits the back of the eye, becomes surrounded by the elevated pressure. The globe of the eye flattens slightly from the normal spherical shape. The optic disc develops edema. The choroid, the vascular layer beneath the retina, develops folds. And vision changes, not dramatically for most people, not catastrophically for most people, but measurably, consistently, progressively. Studies examining astronauts returning from 6-month ISS missions have found that roughly 60% show at least one of the structural signs of SANS. Some develop significant vision changes. Most don't require immediate medical intervention, but the changes are not trivial, and critically, they don't fully reverse in all cases. Research is ongoing to determine whether repeated long-duration missions cause cumulative, irreversible optic nerve and retinal damage. Some data suggests they may. The mechanism matters. The intracranial pressure elevation is not a bug of space travel that can be engineered around with a better spacecraft. It is a consequence of removing the gravity that normally drives fluid distribution. As long as you are in zero gravity or even substantially reduced gravity, cerebrospinal fluid pressure will be elevated relative to Earth levels. The optic nerve will be bathed in elevated pressure. Over months, over years, over decades, the question is not whether some damage occurs. The question is how severe it gets. Several countermeasures are being studied. Lower body negative pressure, a device that draws fluid back toward the lower body by creating a vacuum around the legs, shows promise for reducing the headward fluid shift. Some researchers are investigating special sleeping positions and daily routines that might help redistribute fluid. None of these have been proven adequate for long-duration missions. None of them have been tested over the kind of timescales that an interstellar transit would require. But the eyes are only part of the intracranial story. The brain itself changes in measurable ways during spaceflight. MRI studies of astronauts before and after long-duration missions have found significant changes in brain structure. The cerebrospinal fluid spaces around the brain expand. The brain moves slightly upward in the skull. White matter integrity changes. The volume and shape of specific brain regions alter. Some of these changes appear to be adaptive, the brain reorganizing its processing in response to the novel demands of weightlessness. Some appear to be at least potentially pathological, particularly the changes associated with elevated intracranial pressure. A study published in the New England Journal of Medicine in 2017, examining 15 astronauts using high-resolution MRI, found that most showed evidence of upward displacement of the brain within the skull, along with narrowing of the cerebrospinal fluid spaces in the top of the skull. These changes were consistent with chronic mild intracranial hypertension. They persisted after the missions ended. The long-term neurological consequences of this pattern of change are unknown. For 42 years of transit, the potential consequences are not reassuring. Chronic elevated intracranial pressure is not benign. Over years and decades, it can cause progressive vision loss. It can impair cognitive function. It can cause headache and fatigue. It can accelerate the kind of neurological changes we associate with aging and neurodegeneration. The brain you need to be fully functional when you arrive at your destination, the brain you need to assess an alien environment, make critical decisions, potentially deal with crises that require peak cognitive performance. That brain has been under sustained pressure for decades. There is no reason to believe this is adequately solved by artificial gravity alone, because the question of how to maintain artificial gravity during all phases of a multi-decade mission, including periods of acceleration and deceleration, is itself deeply complex. And there is no reason to believe any countermeasure we currently know of would be sufficient at these timescales. The brain changes are not only structural. The function of the nervous system appears to change in deep space in ways that extend beyond the mechanics of fluid pressure. Research examining the cognitive performance of astronauts on long-duration ISS missions has found changes in spatial cognition, in reaction time, in executive function. NASA's CIPHER project, the complement of integrated protocols for human exploration research, is currently investigating how the brain adapts across missions of different durations, specifically trying to understand what happens as mission lengths increase beyond the current 6-month standard. Preliminary findings, including unpublished mission data from 12-month station stays, suggest that the cognitive effects become more pronounced with longer durations, that the brain does not simply adapt and stabilize, but continues changing in ways that have not reached a plateau. Only 4 humans have ever spent consecutively more than a year in space. The maximum continuous spaceflight is approximately 437 days, set by Russian cosmonaut Valery Polyakov on the Mir space station in 1994 and 1995. He came back to Earth and recovered, though the recovery was extensive and demanding. But 437 days is to 42 years what a morning jog is to running across a continent. We have no data from anywhere near the durations an interstellar mission would require. We have animal data, we have extrapolations, and we have increasingly alarming signals from every direction those extrapolations point. There is something we need to talk about that is rarely mentioned in discussions of interstellar travel, perhaps because it is so intimate and fundamental that it makes the whole enterprise feel uncomfortably human in a context we prefer to think of as purely technological. We need to talk about what space does to the human immune system, because the immune system is not a peripheral concern. It is the defense network that keeps the entire biological machine functioning in a world full of pathogens, rogue cells, and molecular damage, and space is quietly, systematically disabling it. The immune system's dysregulation in spaceflight was documented in early missions and has been studied in increasing detail ever since. The basic picture is consistent. spaceflight alters the balance and function of virtually every component of the immune response. Natural killer cells, the frontline defenders that destroy virus-infected cells and emerging cancer cells, decrease in activity. T-cell function is impaired. Cytokine profiles shift in ways that suggest chronic low-grade inflammation combined with reduced specific immune responses. The bone marrow, which continuously produces the blood cells that constitute the immune army, is affected by both microgravity and radiation, potentially limiting the replenishment of immune cells. The consequences of this are not hypothetical. They have been documented in real missions: skin rashes, respiratory infections, reactivation of latent viruses including Epstein-Barr virus, varicella zoster virus, herpes simplex virus, and cytomegalovirus. These viruses, once you're infected, stay with you for life, held in check by ongoing immune surveillance. In the immunosuppressed environment of spaceflight, that surveillance weakens and dormant viruses wake up. On a short mission with medical support available via rapid return and communication with ground-based physicians in real time, this is manageable. On a multi-year interstellar transit with no possibility of emergency medical evacuation and communication delays measured in years, an outbreak of a reactivated pathogen among an immunocompromised crew could be mission-ending. And this is before we consider the possibility of pathogen evolution. Any living organisms on a closed spacecraft, from the gut microbiome of the crew to any contaminant organisms that made it aboard, will evolve in the pressures of a novel environment. The selective pressures of a spacecraft, the UV light used for sterilization, the antibiotics used to treat infections, the altered immune responses of the crew, will favor microorganisms that resist those pressures. Over decades, the microbial environment of a closed spacecraft might produce organisms significantly more virulent or drug-resistant than those that embarked at the beginning of the mission. The crew's immune systems, already compromised by radiation and microgravity, would be facing an evolving set of pathogens while their own defenses continued to degrade. There is also the microbiome itself to consider. The trillions of bacteria, fungi, archaea, and viruses that live in and on the human body are not passengers. They are partners. They are involved in digestion, in immune education, in the production of essential vitamins, in the regulation of mood and cognition through the gut-brain axis. The composition of the microbiome is profoundly affected by the environment. Studies of ISS crews have shown that the microbiome changes significantly in space. Diversity decreases. Certain species come to dominate. The balance that existed on Earth, shaped by diet, by exercise, by exposure to the outdoor environment, by contact with other human microbiomes, shifts in the artificial closed environment of a spacecraft. Over 40 years in a sealed system with a fixed food supply, without exposure to environmental diversity, without contact with any external microbial community, what would the microbiome of the crew look like? Would it still support health? Would it still provide the immune education and metabolic support that health requires? These are open questions with no definitive answers, because no one has studied a human microbiome in a completely closed system over anything like that duration. But the trends that have been observed toward reduced diversity and disrupted balance are not encouraging. Let us leave the purely physiological for a moment and enter territory that is simultaneously the most human and the most complex dimension of this problem. Let's talk about what happens to the mind. Not the brain as a physical organ, though we have already discussed that. The mind, the consciousness that lives in that organ, the self that is built from memory, relationship, hope, purpose, identity. Because an interstellar mission does not just place human bodies in space, it places human beings in space. Whole persons with inner lives, with the need for meaning, with the social apparatus that evolution built for a species that has never, in its entire history, been this alone. Isolation is not merely uncomfortable for humans, it is, at sufficient depth and duration, literally lethal. research on solitary confinement in prisons, widely regarded as a form of torture under international human rights law, shows that within weeks of total isolation, humans develop symptoms that resemble severe psychosis, hallucinations, paranoia, cognitive disintegration, temporal disorientation, profound depression. These are not mere sadness or boredom. They are the symptoms of a system failing, a mind that requires social input the way lungs require oxygen, beginning to suffocate. The crew of an interstellar mission would not be in solitary confinement. They would be with each other, but they would be isolated from everything else, from the world they came from the families and friends and cultures and languages that shaped them, from the sensory diversity of a natural environment, from the seasons, from weather, from the feeling of soil or sand beneath their feet, from the smell of rain, from birdsong, from wind, from all the vast, uncontrolled richness of an environment that did not understand them and owed them nothing, which is precisely the kind of environment that human brains require for healthy functioning. The research on psychological effects of long-duration space missions and their analogues, Antarctic winter overstations, submarines, isolated research facilities, all points to consistent patterns. The crew initially bonds through the novelty and excitement of the mission. They feel a sense of purpose and privilege. They draw on their extraordinary selection and training to maintain professional focus. And then, somewhere in the middle of the mission, the thing researchers call the third quarter phenomenon happens. Morale drops, conflict increases, sleep disturbances emerge, motivation declines. A kind of psychological middle distance forms in which the beginning feels far away and the end feels farther still, and the confinement of the environment begins pressing in on consciousness in ways that training alone cannot entirely counter. Irritability, social withdrawal, emotional dysregulation, not in dramatic, mission-ending ways usually, but in ways that erode the interpersonal fabric that makes small groups function. Astronauts on the ISS experience this across standard 6-month missions. The Russian space station program documented it extensively in longer missions on Mir. Antarctic winter over personnel have been studied for decades. The patterns are consistent. Social cohesion in small, isolated groups under sustained stress follows a predictable arc that includes periods of significant dysfunction. Now extend that arc over 42 years. Not a 6-month mission, not a 12-month mission, not even the longest human spaceflight on record at 437 days, 42 years, in the same small group of people, in the same limited physical space, with no possibility of relief, no possibility of rotation, no possibility of going outside, with no natural environment, with no contact with the rest of humanity, except for messages that take years to travel in each direction, so that by the time you receive a reply to a message you sent, the world has changed, and the relationships have aged, and the children you left behind have grown up, and perhaps have children of their own. At some point, the question stops being one of psychology and becomes one of identity. Who are you after 42 years? In that environment, are you still the person who left Earth? Is the self continuous across that kind of time, under those conditions? The philosophical questions here are not rhetorical, they matter for the mission. Because if the psychological integrity of crew members degrades over the duration of the voyage, if the social cohesion of the crew collapses, if conflict becomes chronic and dysfunction becomes the baseline, then the mission fails even if the physical systems continue to function. You might arrive at Proxima Centauri with living bodies and a functional spacecraft and a crew that is completely unable to carry out any meaningful activity. There is also the question of purpose maintenance. Purpose is a fundamental human need, as basic as food and sleep according to a substantial body of psychological research. The purpose of an interstellar mission is grand in conception, but monotonous in execution. Years and decades of systems maintenance, of the same routines, of growing the same food and recycling the same water and running the same exercise protocols and sleeping in the same bunk and eating with the same 4 or 5 people who are also, by virtue of the compressed environment, both your closest intimates and your only possible source of social conflict. What does purpose mean in that context? How do you sustain it? How do you prevent the slow erosion of will that comes when the goal is so far away and the daily reality so relentlessly small? Space psychology as a field has been working on these questions with increasing urgency as deep space mission planning has moved from thought experiment to active project development at NASA and other agencies. There are no solved answers. The best current countermeasures, autonomy in scheduling, virtual reality environments, regular contact with Earth, however delayed, deliberately designed privacy, exercise and community rituals, are meaningful improvements over doing nothing. They are not solutions. They are adaptations around a fundamental problem that does not go away. Humans are social, environmental, embodied creatures, and a multi-decade interstellar spacecraft is the most extreme deviation from the conditions required for human flourishing that has ever been seriously contemplated. We have not yet mentioned something so intimate, so fundamental, that it is easy to overlook because it feels too personal to discuss in the context of engineering and physics. But it cannot be overlooked, because if interstellar travel ever moves beyond the single-generation mission concept, if we consider generation ships or multi-decade missions where crew members might have children during transit, we have to confront what space does to the most fundamental act of biological continuity. We have to talk about reproduction. And the answer is, we have absolutely no idea whether human reproduction in space is even possible, and what preliminary evidence exists is deeply worrying. On Earth, human reproduction is an extraordinarily complex biological event. The development of a new human from a single fertilized cell into a functional organism involves a cascade of precisely timed genetic programs, hormonal signals, structural developments, and environmental cues. Every stage is calibrated for Earth's conditions. Gravity plays a role in the organization of cells during development. The absence of gravity, or even the presence of significantly altered gravity, could affect the development of the embryo in ways we do not yet understand. We know that reproduction in microgravity is difficult for non-human organisms. Medica fish have been bred aboard the ISS, but the development of fish embryos showed altered characteristics. Rat reproduction in microgravity has been studied, with findings suggesting impaired fertilization, abnormal embryo development, and reduced litter sizes. The maternal blood volume reduction that occurs in weightlessness creates physiological states that on Earth would be incompatible with sustaining a pregnancy. These are animal models, not humans, but they are warning signals, and they have not been answered with subsequent reassuring human data, for the obvious reason that no one has attempted human reproduction in space. We also know that space affects the hormonal systems involved in reproduction. Testosterone levels decrease in male astronauts during long missions. Ovarian cycle regulation changes in female astronauts. Sperm quality in male crew members has been documented to be affected by the combination of radiation and physiological stress of spaceflight. The cumulative radiation dose an interstellar crew would receive is orders of magnitude higher than any ISS crew has experienced, and the gonads are among the most sensitive tissues to radiation damage. A mission of sufficient duration might render crew members infertile before any reproduction was attempted, or might result in significant DNA damage in reproductive cells that would be passed to the next generation. If children were conceived and born in transit, their entire development would occur in an unnatural environment. Bone formation in developing children requires mechanical loading for appropriate density and architecture. Motor development in infants depends on the gravitational context for calibrating the systems that control movement. Vestibular development, the inner ear systems that provide balance and spatial orientation, requires gravitational input that microgravity does not provide. Cardiovascular development may be affected by the absence of normal hydrostatic pressure gradients. Immune system development in infants depends on environmental exposures that a spacecraft cannot provide. Children born and raised in space might not be capable of living on a planet with normal gravity, not because they chose that, not because they were modified, but because their developmental biology unfolded in the wrong physical context. They would be, biologically speaking, a different kind of human, adapted to space, shaped by microgravity, perhaps capable of functioning in that environment while being unable to survive on the world their parents came from. This is not science fiction speculation. It follows directly from what we understand about developmental biology and the role of environment in shaping the body. Given that we know gravity shapes bone architecture and muscle development, and we know that these processes are most sensitive during development, the implication that development in reduced gravity produces a body adapted to that reduced gravity is not a speculative leap. It is a logical extension of well-established developmental biology. And then there is the question of genetic diversity. An interstellar mission is necessarily a small group. Even optimistically, a generation ship might carry a few hundred people. A few hundred people is, from the perspective of population genetics, a dangerously small founder population. The entire range of human genetic diversity is captured in groups of about 500 individuals approximately. Below that number, you begin to see genetic drift, the random elimination of alleles that do not provide immediate selective advantage, and inbreeding depression, the expression of recessive harmful variants that are normally hidden in a large, diverse population. Over multiple generations in a small closed population, the genetic health of the community would degrade, not quickly and not catastrophically in a single generation, but progressively in ways that compound over time. Diseases that are rare in large populations because the harmful variants are diluted across many individuals would become more common. The genetic diversity that provides resilience against novel pathogens, against novel environmental conditions, against the biological surprises of a destination we know nothing about, would narrow. This is not a theoretical concern. It is the documented consequence of every small founding population in human history. Island populations, endangered species, religious isolate communities. The pattern is consistent. Small populations cut off from genetic exchange with the broader human community suffer genetic impoverishment over generations. The children of an interstellar mission, the grandchildren, the generations that might finally arrive at the destination, would be biologically different from the humans who left, less diverse, more susceptible to specific genetic diseases, potentially less adaptable to environmental challenges. And when they arrive, what will they find? Will the destination be habitable? Will its gravity, its atmospheric composition, its radiation environment, its temperature, its biochemistry be compatible with human survival? Will the world they evolved toward in their closed capsule be anything like the world they encounter? We genuinely do not know the answers to any of these questions. Let us take a step back from the specifics and consider the aggregate picture we have built. We have a species that evolved for one specific planet. That species possesses a biology that begins to malfunction immediately upon removal from that planet's conditions, and malfunctions more severely the longer it remains away. The musculoskeletal system decays in the absence of gravity. The cardiovascular system deconditions. The eyes and brain suffer structural changes from fluid redistribution. The immune system weakens and becomes dysregulated. The genome accumulates radiation damage that is cumulative and only partially repairable. The mind erodes under the weight of isolation and confinement. The reproductive machinery may be compromised by both radiation and physiological stress. And all of this, every single one of these processes gets worse with time. There is no dimension of human biology that improves with extended spaceflight. Not one. Every biological system that has been studied in long-duration missions shows some form of degradation. Some systems show partial recovery after return to Earth. Some show persistent changes that do not recover, and all of them are being studied at timescales that are trivially short compared to an interstellar transit. The nearest star is 4.24 light-years away. With the absolute fastest propulsion system we have today, the Parker Solar Probe, at its maximum speed of about 430,000 miles per hour, a journey to Proxima Centauri would take approximately 6,700 years, not 42 years, not with current technology, 6,700 years. At that speed, no biological countermeasure, no matter how advanced, could keep a human organism functional. You would not arrive. You would not even be recognizable as human by the time you arrived, in whatever state of biological ruin the intervening millennia had produced. Even with hypothetical future propulsion systems that current physics allows for in principle, the numbers remain devastating. A fusion drive, which does not yet exist as a functional spacecraft propulsion system, might eventually achieve a few percent of light speed. At 5% light speed, Proxima Centauri is an 84-year journey, a full human lifetime. If you launched at 25, you would arrive at 109 if you were still alive, in a state of physical deterioration that makes current ISS return conditions look trivial by comparison. At 10% light speed, which would require propulsion technology significantly beyond any currently existing or near-term development, the journey is 42 years, still longer than most human careers, still requiring the astronaut to spend their entire adult working life in the conditions we have described, still biologically devastating under any realistic assessment of the accumulated damage. At 20% light speed, which approaches the theoretical maximum for any beam-driven light sail of the type being explored by Breakthrough Starshot, the journey is 21 years. Time dilation begins to matter slightly at this speed, shaving a few years off the traveler's experience time. But the biological damage from 2 decades of deep space radiation exposure, 2 decades of microgravity effects, however partially mitigated by artificial gravity, 2 decades of immune suppression, 2 decades of psychological isolation, remains utterly staggering. You would not arrive functional. You might arrive alive if everything went perfectly and there were no medical crises and no system failures, and the artificial gravity worked perfectly and the shielding was miraculously effective. But you would arrive profoundly damaged, probably incapable of the kind of physical and cognitive performance that exploring an alien planet would demand. And Breakthrough Starshot's laser sail design, the most ambitious and technically coherent near-term interstellar probe concept, is designed to carry a gram-scale payload. 1 gram. Not a human being, not a crew of astronauts. A gram. Because the energy required to accelerate any significant mass to 20% light speed is beyond any energy system we know how to build. The calculations for accelerating a crewed spacecraft to 20% light speed produce energy requirements that dwarf the total energy output of human civilization. This is the heart of the matter. This is where the biological problem and the physics problem converge into something that looks not just difficult, but structurally impossible, with any technology that obeys the laws of physics as we understand them. We cannot get there fast enough to prevent the biology from failing, and we cannot carry enough shielding and life support to keep the biology intact long enough to travel at any speed we can achieve. The biology and the physics are each independently prohibitive. Together they form a barrier that is not simply a high wall to climb, they form a barrier that exists at the level of physical law. But let's be careful here, because the conclusion that interstellar travel is impossible is only true for a specific kind of interstellar travel. For biological humans, traveling to another star in a single lifetime with no modification to their biology and no modification to the laws of physics. That claim appears to be correct and supported thoroughly by the data we have examined. The more interesting question, and the one that the long arc of time makes genuinely fascinating, is what does interstellar travel become possible for? And at what cost to the meaning of the word human? Consider the problem differently. The biological constraints we have discussed are not arbitrary cruelties of the universe. They are the predictable consequences of applying an organism adapted to one environment to a radically different environment. The solution, in its most basic form, is to change one of those two things. Either you change the environment to better match the organism, or you change the organism to better match the environment. Changing the environment to match the organism more completely is what artificial gravity, radiation shielding, and closed-loop life support attempt to do. We have discussed the limitations of those approaches. They are real and significant, but they are not, in principle, unlimited. Given enough time and enough technological development, a spacecraft could potentially provide an environment similar enough to Earth's that the most acute biological problems were reduced to manageable levels. The shielding problems might be addressed by active magnetic shielding, by creating an artificial magnetosphere around the spacecraft using superconducting magnets carrying enormous currents. This technology does not exist, but it does not violate physics. It would require more power than any current spacecraft can generate, but the principle is sound. The artificial gravity problem might be solved by a sufficiently large, rotating habitat. The psychological isolation problem might be addressed by a large enough crew, by rich enough virtual environments, by social structures designed with all the wisdom of psychology and anthropology. These are not tomorrow's solutions. They might not be the next century's solutions, but they are solutions that do not require abandoning the idea of biological humans in space. They require a civilization vastly more capable than our own, with energy production and engineering capabilities that dwarf anything we currently possess. They require, perhaps, a Type 2 civilization in the Kardashev sense, one that can harness the full energy output of its star. That is a civilization we are not and may not become. Changing the organism is the other approach. This is where things get genuinely fascinating and genuinely troubling in equal measure. The human genome is not fixed. It has never been fixed. For billions of years, evolution has been modifying it in response to environmental pressures. The difference is that evolution operates on the timescale of millions of generations, requiring environmental pressures to be sustained across geological time and survival to be differentially affected by genetic variants. You cannot evolve a human to withstand galactic cosmic ray radiation in any humanly relevant time frame through natural selection. But genetic engineering operates on different timescales. CRISPR-Cas9 and its successors can edit specific sequences in specific cells, not yet in all cells of an adult human, not yet with the precision and safety required for germline modification that would be passed to children. But these capabilities are advancing rapidly, and the trajectory points toward increasingly precise and comprehensive genetic modification becoming technically feasible within decades to centuries. Geneticist Christopher Mason, in his 2021 book, The Next 500 Years, laid out a long-term roadmap for what he calls engineering life to reach new worlds. The proposal involves identifying organisms that already show enhanced resistance to radiation, like Deinococcus radiodurans, a bacterium that can survive radiation doses 3,000 times higher than would be lethal to a human, and understanding the genetic and protein mechanisms responsible for that resilience. Then, over successive generations and technological development, introducing aspects of those mechanisms into the human genome. Deinococcus radiodurans survives extreme radiation through an extraordinary suite of DNA repair mechanisms. It can reassemble a completely shattered genome with hundreds of double-strand breaks within hours. The molecular machinery involved in this repair is well understood and involves proteins that have no human equivalents. Incorporating this kind of machinery into human cells would require modifications at a scale and complexity far beyond anything currently possible. But the principle is not forbidden by biology. Life does this. The question is whether we can engineer it deliberately. Similarly, the tardigrade, that microscopic water bear that survives vacuum, extreme radiation, freezing, and desiccation through its unique set of stress response proteins has provided researchers with genetic and protein tools that might be applied to making human cells more resilient. Research groups have already introduced tardigrade stress response proteins into human cell cultures and observed increased radiation resistance. The step from cell culture to whole organism to heritable modification is enormous, but it is not a step that falls off a cliff of physical impossibility. It is a step that falls off a cliff of technological complexity, which is a different kind of barrier. Bone loss and muscle atrophy might be addressed through modifications to the genes controlling the response of osteoblasts and osteoclasts to mechanical loading signals. The entire signaling cascade that leads to bone resorption in microgravity is understood in considerable detail. Pharmaceutical interventions, bisphosphonates and related drugs, can reduce bone loss in space. Genetic modifications that make this response more robust, that reduce the sensitivity of the bone remodeling system to the absence of gravitational loading, are conceptually feasible, even if practically distant. Cardiovascular deconditioning, the spaceflight-associated neuroocular syndrome, the immune dysregulation, even aspects of the psychological challenge, all potentially amenable to biological modification given sufficient understanding and sufficient capability. The body that would survive an interstellar transit is not an impossibility of physics. It is an impossibility of the biology we currently have. But here we must be honest about what we mean. The human modified to survive 42 years of deep space transit with radiation-hardened DNA repair systems and modified bone metabolism and altered fluid distribution and enhanced immune function and reconfigured psychological stress responses, is that still a human? What does the word mean when the genome has been substantially rewritten? When the biological capabilities and perhaps the psychological experience differ fundamentally from anything that has existed before, this is not a reason to dismiss the possibility. It is a reason to be clear-eyed about what we are actually discussing. We are not discussing sending humans to the stars. We are discussing engineering something new from the biological raw material of humanity and sending that new thing to the stars. The identity question is genuine. If you replace enough of the hardware, at what point is the ship of Theseus no longer the same ship? And the timeline. If genetic engineering capable of producing radiation-hardened, microgravity-adapted, psychologically resilient, interstellar-capable humans requires centuries of development, then the civilization that achieves interstellar travel is not our civilization. It is a descendant civilization that has accumulated technological and biological knowledge we cannot currently imagine. Those people will make their own choices about whether to go to the stars, and they will have capabilities we cannot predict. The question of whether interstellar travel is possible for them is not answerable by us now. It depends on physics and biology and technology that does not yet exist. What we can say with confidence is that for us now, with the biology we have and the technology we have, interstellar travel of the biological humankind is not merely difficult. It is not on the horizon of our current trajectory. The barriers are not primarily technological in the sense that we are a few decades of engineering from solving them. They are biological and physical in a way that is more fundamental than that. There is a third pathway that deserves serious discussion, not modifying the biology, not waiting for physics to produce magic propulsion, abandoning the biological premise entirely. The most efficient way to cross interstellar distances is not with a human body, it is with a mind. Specifically, it is with the pattern of information that constitutes a mind, transferred to a substrate that does not require air, water, food, gravity, or protection from radiation. A substrate that can be replicated, backed up, transmitted, and suspended. A substrate that does not age, does not forget, does not suffer from intracranial pressure or bone loss or immune dysfunction. This idea goes by various names. mind uploading, whole brain emulation, digital consciousness. The concept is that consciousness, identity, memory, all the subjective qualities of being a particular person, are patterns of information encoded in the structure and activity of the brain. If that is true, and it is a contested but scientifically serious proposition rather than mere speculation, then in principle those patterns could be mapped completely and implemented in a different medium. A computer, a distributed system of computing nodes, a substrate that can function in the environment of deep space without the cascading failures we have spent this discussion cataloguing. A mind upload on a spacecraft traveling to Proxima Centauri at, say, 1% of light speed, a speed achievable with sufficiently advanced propulsion, would take approximately 424 years. From the perspective of the uploaded mind, if the system can be placed in a low computation state during transit, this might be experienced as nothing at all, a jump from departure to arrival with no intervening experience. The digital traveler departs, a switch is flipped, and they are at their destination. No 40 years of isolation. No radiation damage accumulating in tissue that does not exist. No bone loss in bones that have not been brought. No psychological deterioration from confinement. No immune suppression. Is this interstellar travel? Philosophically, it depends entirely on whether you accept the identity claims that digital consciousness would require. If you believe, as many philosophers of mind do not, that the subjective experience of a brain can be fully preserved in a different computational substrate, then yes. If you believe that there is something about the physical substrate of a biological brain that is essential to the continuity of consciousness, then the digital traveler is not the person who uploaded. They are an extraordinarily detailed copy of that person who is meanwhile dead. These questions do not have scientific answers yet, because we do not yet have a scientific understanding of consciousness sufficient to adjudicate them. The hard problem of consciousness, why any physical process gives rise to subjective experience at all, remains genuinely unsolved. The debate between those who believe consciousness is substrate independent, a pattern that can run on any sufficient information processing system, and those who believe it requires specific biological implementation, is not a debate being won by either side. But setting aside the philosophical question for a moment and considering the pure pragmatic possibility, digital consciousness as an interstellar travel strategy does not require us to solve the biology problem. It requires us to solve a completely different problem: the full mapping of human neural connectivity and dynamics at sufficient resolution to reconstruct the pattern elsewhere. This is called the connectome, and the human connectome at the level of individual synapses would involve roughly 100 trillion connections, each with specific molecular properties that may matter for the information they store. We cannot map it today. We cannot do so at any resolution remotely sufficient for identity preservation. The Human Connectome project has mapped the large-scale structure of the human brain in impressive detail, but the scale of individual synaptic connections is many orders of magnitude below what we can currently resolve across the whole brain. This technology, if it ever becomes possible, may require centuries of development. It may never be possible in any form that preserves the identity claims we want to make about it. But it is a pathway that circumvents the biology problem entirely, and for that reason alone, it deserves to be taken seriously as part of the long-term thinking about what interstellar travel actually looks like. There is also the option that predates mind uploading as a technology by decades in the science fiction imagination, and that has been discussed seriously by roboticists and artificial intelligence researchers for longer. The robotic probe, the artificial intelligence exploration vehicle, a machine built to be radiation-hardened, to function in microgravity, to survive decades or centuries of transit without food or water or psychological support, designed to explore an alien system and report back. Not a human stand-in, not a digital human, but an authentic artificial intelligence designed specifically for the task. We have already done this in primitive form. The Voyager probes have been traveling for nearly 50 years and are in interstellar space right now. The New Horizons probe has crossed the heliopause. Mars rovers have operated for years on another planet. These are not intelligent systems. They are not capable of the kind of adaptive, open-ended exploration that discovering a potentially inhabited exoplanet would require. But they are proof of a principle. Machines can survive in space for decades. Machines can be designed to function without biological support systems. Machines can operate autonomously in environments where their human creators cannot follow. The obvious limitation of robotic probes is that they are not us. They do not share our experience. They do not carry our curiosity or our wonder or our sense of what it means to be in a place. The first encounter with another world, if one is ever found, would be mediated through sensors and data transmissions rather than through human perception and human presence. Whether that is sufficient is not a technological question. It is a philosophical and emotional one. Whether the discovery of life on another world means more if a human eyes see it than if a camera records it. Whether the expansion of humanity's presence in the universe requires physical human bodies, or whether it can be accomplished through artifacts and transmissions. These questions do not have wrong answers. Different people will answer them differently, and both the answers and the implications are worth taking seriously. We should spend some time with a concept that rarely gets the attention it deserves in these conversations because it is both deeply uncomfortable and genuinely important. The concept is this: even setting aside all the biological and physical barriers we have discussed, interstellar travel of the generation ship variety involves a moral problem that no amount of engineering can solve. It involves children. Not children who choose to go on an interstellar mission, children who are born on one, people who never made the decision to leave Earth, who had no say in whether they wanted to spend their lives in a tin can crossing the dark between stars, who never experienced a natural sky or a natural environment or any of the things that the humans who built the ship valued enough to want to spread across the cosmos. Children who will be born into confinement, who will grow up in confinement, who will live their entire lives in confinement, and who will die in confinement, still in transit, still years or decades from a destination they were pointed toward without consent. This is not a new problem in human history. Children have always been born into circumstances they did not choose, born into poverty, into war, into environments that constrained their choices and limited their possibilities. We do not generally consider this morally acceptable, or at least we aspire to do better. We have developed concepts of children's rights precisely because children are persons, not instruments of their parents' choices, and their welfare deserves separate consideration from the welfare of the adults who brought them into existence. An interstellar generation ship raises these questions with unusual clarity because the constraint is so total and so permanent. A child born into poverty on Earth can, with luck and effort and support, transcend that circumstance. A child born on a generation ship in deep space cannot transcend their circumstance until the ship arrives at its destination, an event that may occur generations after their birth and certainly will not occur in their personal lifetime if the ship is traveling for a century or more. Philosopher Kim Stanley Robinson, who has thought more carefully about interstellar travel than almost any other author working in the field, has concluded through his novel Aurora that generation ship travel is simply not ethically defensible. His argument is that the generations born in transit have no meaningful choice in the matter, and that sufficiently long transit times produce cultural and biological drift that makes the concept of a continuous community, much less a mission with coherent purpose, essentially illusory. The people who arrive, if they arrive, have so little connection to the people who departed that the mission has no genuine continuity of identity or purpose. These are not the conclusions of a pessimist or a Luddite. Robinson is someone who has thought carefully about what space travel would actually involve for real human beings. over real human timescales. His conclusions are informed by biology, by psychology, by sociology, and by basic ethics, and they are conclusions that the scientific community has been arriving at independently, if less poetically. The book Starbound, published by Cambridge University Press in 2025, assembles an extensive analysis of the barriers to interstellar travel and concludes that multi-generational starship travel is, in the words of one reviewing researcher, very, very, very unlikely to succeed. This does not mean that the question of children in space will never arise. If we establish permanent settlements on Mars or in orbit or on the Moon, children will eventually be born there. Those settlements, unlike a generation ship, maintain contact with Earth. They can be resupplied. They can receive medical support. They exist within a range at which rescue is conceivable, and communication is not measured in years. The ethical situation is different, though still complex. But for interstellar missions with transit times measured in decades or generations, the question of the children born in transit is not a peripheral concern to be addressed by a psychologist on the mission team. It is a central moral problem that may have no satisfactory resolution, short of transit times short enough that no children need to be born in transit at all. And achieving transit times that short brings us back to the physics, to the propulsion requirements, to the energy requirements, to the fundamental limits that make the whole enterprise so challenging. There is a way of thinking about this entire problem that offers a kind of dark comfort. A framework that places the difficulty of interstellar travel in its true context, not as a failure of our species or our imagination, but as a feature of the universe that affects every intelligence in it. The Fermi paradox has been mentioned in many conversations about space, and we have touched on it elsewhere in these explorations of the cosmos, but it is worth considering here through the specific lens of biology. The question Fermi asked is simple and devastating. Where is everybody? The universe is 13.8 billion years old. The galaxy is full of stars older than ours. Life, if it arises at the rate we might expect, given the abundance of planets, should have had billions of years to evolve intelligence. And intelligent civilizations, even traveling at modest fractions of light speed, should have spread across the entire galaxy many times over in the time available. And yet, silence. One proposed solution to the Fermi paradox is that interstellar travel is simply very, very hard for biological intelligence. Not impossible in principle, but requiring a level of technological development and a time scale of effort that very few civilizations, perhaps none so far, have actually achieved. That the silence is not absence, it is the sound of everyone being stuck, being constrained by the same biological and physical barriers we have just described, being in the early stages of figuring out how to leave their homeworld in any permanent, meaningful way. This is the great filter in its biological form, not a catastrophic event that destroys civilizations, not a war or a collision or a failure of sustainability, a quiet, chronic filter. The filter of biology, the filter of flesh and bone and blood, trying to traverse a universe that was not built with flesh and bone and blood in mind. If this interpretation is correct, then we are not unusual in our dilemma. We are not exceptional in our difficulty. We are typical. Every civilization that has ever arisen in the galaxy has stared at the same stars and done the same math and arrived at the same uncomfortable conclusion. The nearest star is impossibly far. The body that could stand on its worlds is impossibly fragile. The gulf between what the mind can imagine and what the biology can survive is impossibly wide. And yet here we are, still imagining, still building telescopes, still launching probes, still asking the question. Perhaps that is itself meaningful. Perhaps the imagination that conceives of crossing interstellar space is more significant than any actual crossing. Perhaps the civilization that truly understands the scale of the universe, that looks at the stars with instruments capable of seeing exoplanets, that measures the radiation environments of deep space and calculates the biological consequences, that confronts the gap between aspiration and physical reality with clear eyes, has achieved something more important than any physical journey. We know where we are, we know how small we are, we know how fragile we are, we know how vast the distances are, we know how insufficient our bodies are for the cosmos we find ourselves in. And we know all of this because we have the intelligence, the curiosity, and the persistence to have measured it. That is something. But let's not end there, because ending with resignation would misrepresent the nature of the problem, and misrepresenting the nature of the problem would be a disservice to the people working on it seriously. The barriers to interstellar travel are real. The biology is genuinely problematic in the ways we have described. The physics is genuinely limiting in the ways we have described. Neither set of barriers is going away. They are not going to be wished away by enthusiasm or engineering away by the next generation of rockets or solved away by any near-term development. But they might, over sufficiently long timescales and with sufficiently advanced development, be reduced to manageable levels. Not for us. Not for our children. Perhaps not for many generations after them. But in the long arc of time that a species willing to think carefully about its own future has to work with, the barriers are not infinite. The radiation problem is the deepest, the most fundamental, the most resistant to conventional engineering approaches. But active magnetic shielding, generating a magnetosphere around a spacecraft using superconducting cables carrying millions of amperes, has been studied seriously. At sufficient power levels, this could deflect a significant fraction of galactic, cosmic ray, heavy nuclei, not all of them. The most energetic particles, the ones above a certain energy threshold, will punch through any magnetic field you can realistically generate. But reducing the dose significantly, combined with other countermeasures, might be achievable with energy generation capabilities well beyond ours, but not fundamentally different in kind. Genetic modification of human cells to enhance DNA repair efficiency, inspired by naturally radio-resistant organisms, is active research now. The pace of progress in genetic engineering over the past decade has been genuinely remarkable. CRISPR-based tools have compressed what was expected to take generations of development into years. The path from lab observation to practical germline modification is long and ethically fraught, but it is not invisible from here. The biological machinery required to improve radiation resilience exists in nature. We are increasingly capable of understanding and eventually replicating it. The microgravity problem, the bone and muscle and cardiovascular deterioration, is in principle solvable by adequate artificial gravity. The reason artificial gravity hasn't been used on the ISS or any crewed spacecraft to date is primarily cost and engineering complexity, not physical impossibility. A sufficiently large, well-funded, long-term space program could design spacecraft with rotating habitats that provide Earth-equivalent gravity. This is more than a thought experiment. It is a serious engineering challenge with known solutions at sufficient scale and energy. The Herculean cost is the barrier, not the physics. The psychological challenge is, perhaps unexpectedly, the most tractable of the serious problems in some respects. Not because it is simple, but because human beings are remarkably adaptable when they have adequate support, social structure, and purpose. The psychological problems documented in long-duration missions are real, but they are documented and understood and actively researched. Interventions exist that reduce the severity of isolation effects. Better crew selection, better social design, richer virtual environments, more autonomy, more connection to the mission's meaning through continuous scientific activity, all of these improve outcomes. A sufficiently well-designed mission with a crew large enough for genuine social diversity and a social structure thoughtfully designed around what we know about small, isolated groups would have better psychological outcomes than the missions we have run so far. Not good outcomes necessarily, but better. And there is the wild card of technology we cannot yet predict, just as the 18th century natural philosopher could not predict the semiconductor or the radio telescope or the molecular biology that would eventually let us read the genome. We cannot predict the discoveries of the 22nd or 23rd century. The history of science is a history of surprise. The laws of physics as we understand them do not promise magic. They do not promise warp drives or wormholes. But they do not promise that the current state of knowledge is the final state. There are things we do not yet know. What we can say with confidence is that the gap between where we are and where we would need to be is not measured in decades or even centuries if we are talking about biological humans traveling to another star. It is measured in something longer, in the kind of time that requires us to think about ourselves as a species on a civilizational arc, not as individuals making career plans. In the kind of time that requires humility about what we are and what we are not yet, and patience with the long, uncertain path from here to there. Here is something worth sitting with. The Voyager 1 spacecraft, launched in 1977, is currently in interstellar space. It has been traveling for nearly 50 years. It has covered approximately 160 astronomical units, roughly 14.8 billion miles. It is, by any measure, the most distant human-made object in existence. It carries the golden record, that extraordinary capsule of Earth sounds and images and greetings in 55 languages that Carl Sagan and his team composed as a message to whoever might find it. Voyager is in interstellar space, but it is not near another star. The nearest star, Proxima Centauri, is 268,000 astronomical units from Earth. Voyager has covered approximately 160 of those. After 50 years of travel, it has completed roughly 0.6% of the distance to our nearest stellar neighbor. At its current speed, it would take approximately 73,000 years to reach Proxima Centauri if it were heading in that direction, which it is not. This is not a failure. Voyager is a triumph of human engineering, operating beyond all expectations, returning data from a place no object we have made has ever been before. But it puts the problem in perspective that no abstraction can quite achieve. We have been traveling for 50 years. We are nowhere near another star. The fastest thing we have ever launched is nowhere near another star. Every human who had anything to do with the Voyager program has retired or passed away, and the spacecraft has barely started its journey. The body that could make this trip does not exist. The physics that could make it fast enough to matter for living organisms does not exist in deployable form. The spacecraft that could protect a crew from the environment they would traverse does not exist. These are not failures of will or imagination. They are the consequences of physics and biology and the brutal arithmetic of cosmic distance. And yet, the fact that we can describe the problem this clearly, can name every barrier, can identify every physiological system that would fail and understand why it would fail, is itself a kind of progress. The first step towards solving a problem is being honest about what the problem actually is. We have spent generations in the mythology of space travel, the Star Trek optimism that technological cleverness would make the cosmos accessible in roughly the way Earth has become accessible by air travel, bold, fast, affordable, reversible. The data says otherwise, the biology says otherwise. What comes after that mythology is not despair, it is clarity. And clarity is where real progress begins. The species that colonizes the stars, if our species or its descendants ever do, will not look like the astronauts of the 20th century in bigger ships. They will not be humans in the biological sense we currently inhabit, traveling at speeds achievable with chemical rockets or even fusion drives, eating food and breathing air, and worrying about their bones. They will be something else, something that emerged from us over centuries of technological and perhaps biological development, something capable of surviving the universe as it actually is, rather than the universe we would prefer it to be. Whether we would recognize them whether they would recognize themselves as us, whether the question of identity and continuity matters at all across those kinds of transformations. These are questions that we cannot answer. They belong to futures we cannot see. What we can say, standing here in this moment with our finite biology and our finite reach and our infinite imagination, is that the stars remain unreachable for us now, not as a judgment, not as a sentence, just as a fact, as clear and as humbling as the void between galaxies. And the fact that we can say it precisely and clearly, that we can measure the distance and describe the damage and name every barrier with the language of verified science, means that we are paying attention, means that we are taking the problem seriously, means that we have decided not to console ourselves with stories that don't survive contact with reality. That is what science is. That is what this kind of honest reckoning with the universe looks like. Not the celebration of what we can do, not the denial of what we cannot, but the clear-eyed examination of both together as a foundation for whatever comes next. Let us come back to the beginning. Your body is destroying itself right now, slowly in the ways that all biological systems tend toward entropy, toward disorder, toward the gradual unwinding of the complex organization that constitutes being alive. On Earth, this process is manageable. The environment supports it. The planet we live on was, in a very real sense, built to sustain us, or rather we were built by it, to be sustained by it. Every organ, every cell, every molecule of you is in a continuous conversation with an environment that has been shaping life for nearly 4 billion years. Remove you from that environment and the conversation becomes increasingly one-sided. Remove you from gravity and your bones dissolve. Remove you from the magnetosphere and your cells are struck by cosmic bullets. Remove you from psychological connection and your mind begins to eat itself. Remove you from genetic diversity and your descendants begin to lose the biological robustness that only large populations maintain. Remove you from the planet that made you and you begin systematically to come apart. That is the fatal flaw. Not in any one system, not in any correctable design error, not in some feature that a clever engineer can fix, in the fundamental mismatch between the biology that evolution produced and the universe that biology would have to traverse. We are Earth creatures, comprehensively, irreducibly, from the calcium in our bones, to the pressure gradients in our blood, to the microbiome in our gut, to the developmental triggers that shape the brain. We are a product of one world, living in a universe of hundreds of billions of galaxies, and the distance between any world and any other world dwarfs every measure of scale that our Earth-adapted minds can intuitively grasp. The cosmos is not hostile to us. It is indifferent to us. It does not know we exist. The radiation that shreds our DNA is not aimed at us. The vacuum that would kill us in seconds is not hostile. It is simply what space is, what the universe between worlds is, what we would have to cross if we wanted to reach anywhere else. And what we are, biologically, is not adequate for that crossing, not in any form or at any speed achievable by our current civilization. This is not the end of the story. It is not even close to the end of the story. The story of how a small, fragile, Earth-adapted biological species looked at the stars and said, "We want to go there," is barely in its opening pages. What comes next? What the next centuries of development in biology and propulsion and artificial intelligence and perhaps in our own biology produces in terms of capability and possibility is genuinely unknown. But it begins from here, from this honest accounting, from looking at the data and saying, yes, the body fails in space. Yes, the radiation is lethal over the required timescales. Yes, the biology is the problem as much as the physics. Yes, reaching the stars in any meaningful sense is far harder than anything we have yet attempted. harder perhaps than anything we can currently imagine attempting, and saying it anyway, writing it down anyway, working on it anyway. Because the fact that we are a species that can conceive of crossing the distance between stars, even while being forced to acknowledge every reason we cannot, is itself something remarkable. The imagination is larger than the biology, the desire is wider than the universe allows, and in that gap between what we are and what we want to reach for, in that permanent, painful, beautiful gap, is everything that makes us what we are. We are Earth creatures. We live at the bottom of an atmosphere, protected by a magnetic field, sustained by a planet that shaped every protein in our bodies. We look up at the stars and we feel something that no asteroid, no gas cloud, no neutron star feels. We feel longing, we feel curiosity, we feel the particular, irrational, irresistible pull of wanting to go to places we cannot reach and see things no human eyes have ever seen. That feeling is not a flaw. It is the most extraordinary product of 4 billion years of biological evolution. The universe has spent billions of years getting to the point where some of its matter looks up at the rest of it and wonders. We are that wondering. We are the cosmos looking at itself and asking, what else is out there? We just need much better bodies to go find out. For now, the stars will wait, and we will keep measuring the distance,